are, well, is there any specificity? So maybe there are some brain regions that are just specifically activating during fear. And then there are other brain regions that are specifically activating during anger, and so on and so forth. So this has been something that's been debated in psychology since the beginning of psychology, really. Even before that, if you look back into mental philosophy, in the 17th and 18th centuries, people debated about the brain basis of emotion, and they asked about, are there specific brain regions that can be localized for specific emotions. So from the popular, for those of, for those of you, even if those of you who've read, the popular media, if I were to nominate one brain region that I might look at related to an emotion, has anyone here read anything that they might suggest a particular brain region that we should look at? Anything? Amygdala, right. So there's been a lot written about the amygdala in relation to fear. And so here is a plot of the proportion of studies, either disgust or anger studies, or disgust or fear, or happy studies, or sadness studies of those emotions, either where people are experiencing emotion or they're perceiving that emotion. And this is the y-axis, the proportion of studies that shows activation in the amygdala during the perception or experience of emotion. And what you can see is that it's almost all emotions. The amygdala is active for almost all emotions. So no specificity there. What's interesting, actually, here, if you look, that the experience, there's no bar here for the experience of fear. And the reason why there's no bar is that these are all proportions that have been thresholded. They're significantly different from 0. So for fear, the proportion of studies active during fear were not significantly different from 0, meaning half of the studies found amygdala activation during fearful experiences and half of them didn't. And this wasn't accounted for by the methods. The methods themselves didn't predict, that were used to evoke fear, didn't really predict when you would see the activation and when not. So, except for the experience of fear, the amygdala is reliably active during emotion. And there are other brain regions that other people have talked about, the anterior insula and disgust, because sensations from the body are supposed to be specifically or particularly important to experiences of disgust. People thought that maybe the insula would be very specific to the experience or perception of disgust, but you see it's not. And then for anger, people have talked about a region of orbital frontal cortex, and again, you see this kind of lack of specificity. So really, these findings tell us, yeah, so before you read this, let me just say this. So one criticism of neuroimaging studies in humans is that the spatial resolution, while it's better than it used to be, is still not great. Meaning, even if you did the most precise imaging experiment that you could, and you divided the brain up into little three-dimensional, they're called voxels, so it's really a three-dimensional pixel, right, is a voxel. Just you take the whole volume of the brain and you divide it up. You get something like 180,000 voxels that are maybe 3 millimeters cubic, or 2, or if you're really using something really sophisticated, 1. Now, how many neurons do you think are in 1 millimeter of the brain? A gazillion. Like, that's a technical term. But I mean, many, right? So one argument is, well, you're not going to see specificity because, your measurement is too coarse. But here's the thing. So my students and I decided, okay, well, we're just going to read then about electrical stimulation studies. These are studies where you implant an electrode in a single neuron, and then you see what happens. You stimulate the neuron, and you see what happens to the behavior. And so there are tons of these studies. Most of these studies were done really in the mid part of the 20th century. Those studies are still being done in very sophisticated ways. But we basically read through the literature, and this is a summary, a quote from Elliot Wallenstein, who was a very noted neuroscientist in his time who did many, many, many electrical stimulation studies. And he wrote, and I just think this is a great summary of because the idea would be that maybe you could find specificity if you were doing single cell recordings. And he wrote, the impression exists that if electrodes are placed in a specific part of the brain, a particular behavior can inevitably be invoked. Those who participate in this research know that this is definitely not the case. Even in rats, where the behavior is more stereotyped than in monkeys and man, brain stimulation produces variable results. The experimental data clearly indicate that electrodes that seem to be in the same brain locus in different animals can often evoke different behaviors, and electrodes located at the very same brain sites can evoke the same behavior in a given animal. So, if you put a rat, a male rat, in a cage, and you drop in a conspecific that the rat doesn't know, he will attack that rat. And you will see an assembly of neurons in subcortical regions that support that attack behavior. If you put, and that's true if it's a subordinate conspecific that you've dropped in, but if you put another rat in who's more dominant, you see the same behavior, but you don't see the same assembly of neurons active. You see, it sort of overlaps, but there are many parts that don't. What does that mean? Or if you take a monkey and you stimulate a particular part of the temporal lobe in a monkey, the monkey might attack, depending on who the other monkeys are, but it might not, depending on who the other monkeys are. And it also matters what the monkey's own temperament is. So what does this say? It's not saying that you can't look at the brain and predict behavior. It's saying that you have to consider other kinds of factors that are, we think of as context factors. So my point here is that fMRI, oftentimes when I show you those pretty, that pretty brain with all those colors, I think I have it here again, people, and I want to make certain claims about the lack of specificity, not just for different emotions, but actually for perception and cognition. People will say, well, but neuroimaging isn't really precise, and they're right, it isn't really precise, but if you look at the most precise methods, you don't find something terribly different. You might find specificity for a specific behavior in a specific context, right? So attack in a specific context, but not attack in another context. That's a slightly different assembly of neurons. So, yeah, let me just back up. So I'm telling you here that there's a lack of specificity, but you can actually, just because emotion and cognition and perception are not the categories that work to parse those data, or anger or sadness or fear, does not mean that there's no organization in those data. So if we take that brain space and we subject it to other kinds of statistical analyses, like multidimensional scaling and cluster analysis, we can look for functional groupings in the human brain. That these are, voxels that are showing the same pattern of activation across different kinds of studies. And what we found is that we found 6 different groupings. And if I had 2 hours to talk to you today, or a whole course even, I could go over with you what these groupings are. But what I want to say today, the take-home message for you today is to not so much worry about what these groupings are, but more for me to say 2 things. One is that these same these groupings look like they have a passing resemblance to what are called intrinsic networks in the brain. So if you look at a human brain in an imaging study at rest, meaning, now the brain is never really at rest, but that's what it's called, at rest, which just means you're not probing the brain with a stimulus. And you look at you take the activation time course in a given voxel, that is, you look at its fluctuations of activation, and you compare that to the time courses of fluctuation in other voxels, you find networks of regions that are widely distributed across the brain that kind of fluctuate, their activation time fluctuates together. And they happen to be more or less anatomically constrained. So what does that mean? It means that your brain is populated with a set of networks. And those networks kind of look like these functional groupings. The second point to make about these functional groupings is that they appear in other kinds of studies. So they appear in studies of memory. They appear in studies of person perception. They appear in studies of object perception. They appear in studies of attention. So the same functional groupings are showing up again and again and again in different kinds of studies, and they're similar to these intrinsic networks that appear to be anatomically constrained. So if we really want to understand how the brain creates the human mind and in general, and specifically creates instances of emotion, the goal is really to figure out what exactly these networks do in psychological terms. Because a brain network only becomes a mental process when you can describe it in terms of the function of a living, breathing human, right? So we need to figure out what are these networks doing and how are they working together to produce the mental states and actions that we all experience every day. And that is really the sort of cutting edge of the intersection of psychology and cognitive neuroscience. So if this is not, these kind of data are not consistent with a psychological model, with a psychological idea that you have different kinds of faculties and there are different brain regions for different faculties, what kind of model is it consistent with? Well, it turns out these data are consistent with a kind of a model that in the psychological literature is called psychological construction. And the idea here is that emotions, like all mental states, arise from a set of basic ingredients. So the mind, you can describe the mind as a well-stocked pantry that can be used to make any number of ingredients, which are the mental states that we give common sense names to, like sadness and anger and beliefs and memories and so on. And so more formally, the hypothesis is that every human brain is populated with large-scale brain networks, and these can be redescribed in psychological terms. And that it's these psychological ingredients, really, that are the basic sort of ingredients that we use to construct mental states and also to communicate with each other about mental states. And the cool thing is that the mental categories that we communicate with each other about and that we teach our children about actually affect the structure of the brain. As it turns out, the brain is a highly plastic organ and can be influenced by social learning and cultural variation. And what this suggests is that you should be able to see cultural variation in imaging studies of basic phenomena, like does fear look the same in people of all cultures? And there are nice neuroimaging studies showing really nice cultural differences that would be very much predicted by this kind of an idea. This kind of an idea is a very old idea in psychology. So William James, who is considered to be one of the forefathers of psychology, certainly of American psychology, wrote in 1890, "A science of the relation of the mind and the brain must show how the elementary ingredients of the former correspond to elementary functions of the latter." And one thing for those of you who are interested in psychology, you will find that anything that you ever read or ever decide to write was probably already said by William James extremely well. So when I started developing my own ideas about emotion, I mean, I don't know, I read William James when I was in graduate school, but he's written a lot of stuff, and you don't remember every single thing you read. And when I came across this quote a couple of years ago, I literally, the book dropped out of my hands. I was like, I can't believe he already said this. I'm sort of despairing that I'm never going to say anything novel. The thing is that even though William James said this actually is an incredibly, it's novel in the 20th and the 21st century, meaning, this kind of a view was not taken seriously for a very long time. And I think now it's taken much more seriously. So the last thing I want to show you is I want to show you that you can uncover these ingredients in a psychology laboratory without looking at a brain by doing careful experimentation. And the experiments I'm going to show you are experiments on emotion perception. So we all have the experience every day. We look at someone's face and instantaneously we know exactly how they feel. We're often wrong, but that's okay. Because the point is that we just do it automatically and effortlessly. It feels to us, like emotions are just displayed on the face there for us to read. And in fact, if you look up in any psychology textbook, you will find a statement about the fact that there are 7 plus or minus 2 emotions which are supposed to be expressed on the face with very characteristic expressions, that everybody makes them and everybody around the world can detect them, and you're born with this ability. Okay? So from that perspective, there should be no basic ingredients of perceiving emotion, because it's it's kind of displayed on the face for you to read, and then you just use your inborn fear detection mechanism and you detect fear. And so here are the experiments. Here's a kind of experiment that has been shown, that's shown these effects. So you show somebody a really caricatured kind of extreme version of an expression, and you give them a bunch of words, and then you say, which word matches that face? So which word matches that face? Anger, right, exactly. And if you do this across cultures, people have pretty high accuracy rates. About 85% of the time, they get it right. Meaning they agree with each other and they also agree with the experimenter. That's what it means to get something right, that you agree with the experimenter. Okay. But what happens if you just give somebody this highly caricatured face and you give them no words? You say, how does that person feel? Accuracy rates drop to 58% within a single culture. And if you remove words from the task even more, so instead of having someone produce a word, you show them 2 faces and you say, do these people feel the same way or not? So you're basically asking them to make a match or a mismatch judgment. This is called a perceptual matching task. So are these 2 stimuli the same or not? So is this person displaying anger? Is this person displaying anger? If so, then you would say yes, right? Okay. And if this person was displaying something else, then you'd say no. If you do that, accuracy actually drops to 42%, which is significantly different, but still different. It's still above chance, but significantly lower than 58%. So basically, the more you remove words from the task, the worse people get at seeing emotion in a face. And there's a laboratory procedure, it's very technical, it's called semantic satiation. Here's how semantic satiation works. You take a word and you repeat it over and over and over again, like 30 times. And when you do, the word sounds like gobbledygook to you. So my daughter discovered this. She's like a pure experimenter. She discovered this. Every child, right, discovers this. It's hysterical. They kill themselves laughing. Except this is actually a scientific procedure we use in the lab. It's been used since the 1970s in cognitive psychology, where you get a person to repeat a word over and over and over. What you do is, what's happening is that the phonological form of the word is being temporarily detached, essentially, from the meaning of the word. So people they hear sounds and they don't know what they mean, essentially. So you're basically reducing the accessibility of emotion words. And when you do that, accuracy on this perceptual matching task drops to 36%, which is a significant drop. And also, it reflects the fact that people can still match on valence. They can still tell you, they can still say when 2 faces basically agree that they look both look negative. But they really, what they can't do is tell you that both faces are scowling faces. And this is, I should point out, so we get people to repeat the word anger 30 times, and then we have to do this task, and they show this reduction. The word anger appears nowhere in this experiment. They don't have to label those faces. They just have to say, do these faces match or not match? So one thing that you might think is, well, okay, so language appears to be important to these tasks. So as long as language is in the experiment, you get cross-cultural agreement on what those faces mean. If you remove language from the experiment, you can't get a bunch of homogeneous undergrads in one culture to agree with each other. Okay? So what appears to be, cross-culturally stable here is that language is shaping the perception of emotion. And actually, I have a graduate student right now who's in Namibia who's testing this hypothesis with people on the Himba tribe, to see whether if language really shaping perception, and it appears to be the case. So I was going to show you another experiment where we can show that, but I'm just going to skip it for time purposes. So I was going to explain to you another kind of method that we use to show that language doesn't just, I tried to skip it over there, but I couldn't do it, so you'll have my apologies. That language doesn't just shape the categorization of emotion after the fact, it actually shapes the construction of an emotional percept. So your ability to see a scowl as angry, depends on your ability to access the meaning of the emotion. You'll just have to trust me. That work is in press, and it's on my website if you want to see it. What I want to say is, you can also look for a natural experiment. For example, you can look at people who have deficits in language processing because of neurodegenerative disorders, so patients with semantic dementia. And if language really is a basic ingredient of emotion perception that we can reveal with experiments, then it should be the case that if you looked at a semantic dementia patient and you asked them to perceive emotion, that they shouldn't be able to do it. Right? So they might be able to tell you affect, like is this face positive or negative or neutral, but they wouldn't be able to actually tell you is it angry or sad or afraid. And so we actually did this experiment where what we did was we gave subjects caricatured faces. So all the angry faces were scowling, all the sad faces were pouting, all the fear faces looked startled, and so on and so forth. And we gave them 6 kinds of faces. And we said, just sort these into piles where everyone in the pile is feeling the same thing. And here's what they do. They sort all the smiling faces into one pile, and also some neutral faces, and one fear face. They sort almost all, actually all the negative faces, almost all of them, into one pile. And then they have neutral faces in another pile. And the neutral faces are distributed across the 3 piles because just the physiognomy of a face, just its structure, actually can make a face look positive or negative or neutral, even if it is not, it has no expression. So these data show that people with semantic dementia can perceive affect, but they can't perceive emotion. And it turns out that if you give them, so instead of having them sort into piles, you actually give them an anger face. So you put down a smiling face, and you put down a scowling face, and a startled face, and so on. And you say to the patients, what I want you to do is to take the target faces and match them to the anchor. They can do it. So it's not that they can't see a scowl as a scowl. Well, it's not that they can't see that the scowl is, no, they don't know it's a scowl. What they know is they can match the perceptual features of a face to another set of faces, but they don't know that the face is a scowl. And they don't know that the face is angry. In fact, one patient even said, well, what's anger? And this is a patient who could sort, she could do that perceptual matching test with scowls. She couldn't label the expression, and she didn't know what it meant psychologically. So if it feels to you like you're reading words on a page when you perceive emotion, you probably are, in these experiments. But more than that, language is this pervasive ingredient that is always functioning in your waking life. You're just unaware of it most of the time, but it's there. And it's affecting emotion perception, and it's probably affecting other things as well. So the take-home message here is that, specifically for emotions, is the idea that emotions are probably not these preserved little circuits in the brain. They're probably mental states that are constructed from more basic psychological ingredients, and that these ingredients appear to correspond to distributed brain networks, which is I think one of the greatest discoveries really in the 21st century about the human brain is the existence of these networks. And the idea is that it's not like your emotions are wild horses and that you're the driver. That's not a good analogy for what's happening in the brain. Instead, a better analogy is that you are a chef, and that you have available to you a set of basic ingredients that are being combined in every instant to produce the thoughts and feelings and actions that you engage in. And this, chef metaphor, I really like it, not only because I think it's a better metaphor, but also it helps us to see how psychology and neuroscience are kind of two sides of the same coin, in the sense that psychology is kind of a description of what the recipes are, and how, what ingredients you need, how you combine them, and so on. Whereas neuroscience is really more like an explanation of how those recipes work. And with that, I will thank you for your attention and take, I don't know if there are any time for questions.